Good morning, everybody, and welcome to the modeling session of this Nanosafety Training School. In this section, Professor Buchete, University College Dublin, Professor Agur Sevink, Leiden University, and myself from Politecnico di Torino are going to give you a taste of which modeling techniques can be used to calculate quantities that are related to toxicological endpoints. The first short lecture is focused on Brownian Dynamics, a computational modeling technique useful to get insights into the nanoparticle aggregation process. As you may know, nanoparticles are potentially toxic because of their small size, which makes it easier for them to penetrate into cells, where they could exert a wide variety of damages. In this first lecture, we will see some example of descriptors for nanomaterials, the multi-scale approach adopted by nanoinformatics work package 3 modeling partners to focus then on classical molecular dynamics and Brownian dynamics as a possible way to upscale molecular dynamics simulations to study larger systems made of thousands of nanoparticles. Descriptors are physicochemical quantities specific of the material under investigation. Descriptors can be calculated using several computational techniques and also experimentally measured. We are talking about nanomaterials, but still the size of the materials themselves and the biological entities in contact with them is too large to be computationally feasible at a quantum scale. However, quantum descriptors represent a useful first principle information specific to each material, which can be used as an input for quantitative structure-activity relationship QSAR models, which are basically mathematical models that can be used to predict the physicochemical, biological, and environmental fate properties of compounds from the knowledge of their chemical structure. Using molecular dynamics, one can see nanoparticle-nanoparticle interaction, nanoparticle-protein, and nanoparticle-lipids interactions, also making use of coarse grained models as those you will see in the following lectures. Surface charge, which is an important quantity for nanoparticle aggregation and interaction with biological moieties, can be calculated using reactive force fields molecular dynamics. Some descriptors are measured experimentally, such as zeta potential, which is the potential difference between the dispersion of the colloidal suspension and the stationary layer of fluid attached to the particle. Zeta potential is related to the stability of the colloidal suspensions and is estimated experimentally through electrophoresis or calculated using some formulas. So the modeling approach is multi-scale in the sense that techniques employing different levels of approximations are used for systems of different sizes. All the calculated descriptors can be fed into QSAR models, which allow predicting toxicological endpoints. The calculation of descriptors is generally quite expensive in terms of computing time, so it is harder to generate a large dataset. Moreover, the quantities are very specific to the input structure. For example, they depend on the size and also on the shape of the nanoparticle simulated, its crystalline or amorphous structure, and the results are sometimes hard to compare with the experimental ones because the computational conditions are somewhat perfect. For example, a perfectly spherical nanoparticle in pure water, whereas the experimental conditions are more complex. There are ions in solution, proteins, chelating agents, and also the exact size distribution of nanoparticles may be unknown, as well as the coating information may be lacking. Despite these limitations, computational calculated descriptors are valuable and represent a good starting point for toxicological evaluation based on the chemical structure. Nanoparticle aggregation changes the size and shape of the material, determining its cellular uptake and therefore its toxic effect. I calculated the aggregation free energy for pairs of identical nanoparticles in water, which is the main solvent in body compartments. Let's see now how to calculate the aggregation free energy using molecular dynamics. So I used the DL Poly software with the force field of Matsui and Akaoji, widely used since its development in 1991. Here you can see the input parameter for the simulations. I created a cubic box with a nanoparticle in the center. Then I filled the box with water and I replicated it along X to get a final orthorhombic box with two identical nanoparticles surrounded by water. After an initial equilibration in an NV and in an NPT ensemble to mess up the initial configuration of water molecules and also to adjust the density, I started to pull the two nanoparticles towards each other. This normally would take a very long time, well, actually short for titanium dioxide, as we will see later, but very, very long for the classical molecular dynamics time span. So an enhanced sampling technique must be used. I choose the umbrella sampling, which is a technique discovered in the 70s, which consists of a harmonic potential used as a driving force for a chemical phenomenon. The first thing to do is to choose the proper reaction coordinate to get the right information. In this case, I want to get the free energy for the aggregation of two nanoparticles at a specific distance. To do so, I set as a reaction coordinate the center of mass distance. I divide this distance the nanoparticles have to cross into target distances. I set up a molecular dynamics simulation at each of these target distances with the help of a potential with a harmonic constant K. I used a DL Poly version patched with the Plumed tool, which is a very useful software to perform any kind of accelerated sampling techniques. In the right-hand side of this slide, I reported the oscillation of the center-center distance during each umbrella sampling simulation. You can see that the oscillations around the target value overlap, and this is fundamental for combining the simulations with the weighted histogram analysis. Basically, this analysis works on the principle that if you have a discrete number of states, you can create a histogram with the discrete beams that provide you a relative probability of observing the states of interest, assuming that you create the beams along the reaction path of your choice. In this slide, I reported the free energy for the aggregation of two nanoparticles in water. The depth of the potential well gives the aggregation free energy, but the Wool curve is interesting as it can be fitted by mathematical equations describing the repulsion at a short distance and the attraction at longer distances, as I will show you in the next slides. In this specific slide, you can see the free energy profile of a 4 nanometer diameter nanoparticle of titanium dioxide anatase as an example. The aggregation free energy here is about 17 kilocalories per mole. In the first molecular dynamics simulation after equilibration, the two nanoparticles are placed at around 6 nanometer, considering the center-center distance. You can see that the free energy decreases slightly as particles get closer, and the minimum is at a distance of 4.4 nanometer. Closer than this distance, nanoparticles start to repel each other. As a distance is lower than 4 nanometer center-center, they would compenetrate. So we have now the first descriptor, the aggregation free energy. And let's see how to upscale using this information. So in classical molecular dynamics, one uses a force field, which is a set of mathematical equations and their parameters obtained fitting quantum calculations or experimental data. This set of equations describes all the interactions between the atomic species present in the system. Now we want to use the information previously obtained using one of these force fields to see the aggregation of many particles. To do so, we assume that each spherical nanoparticle can be represented by a bed, which is a rigid sphere with the diameter equal to that of the nanoparticle. Then we create a custom potential fitting our free energy curve. In this way, each bed will interact with other beds according to this new potential, which takes into account the effect of water, because this was derived in explicit water. So GROMACS 2019 software allows specifying a custom potential, but unfortunately the newest version doesn't support it. But the GROMACS support says that this will be implemented again in the future versions. The way to use a custom potential is to provide as an input a table like this one in this slide, which is obviously truncated. The first column is for the distance x between the particles. The second and the third for a function f and its negative derivative. The fourth and fifth for a function g and its negative derivative. And the last two for a function h and the negative derivative. So F is the electrostatic term, G is the attractive term, and H is the repulsive term. This is why here you only see numbers in the last two columns in H, because at short distances H is the only term. In this case, stoichiometric nanoparticles are charged at 0, so F is also set as 0. The equations can be polynomial or of different orders or even logarithmic, and you use the best fit for your free energy curve, basically. So I prepared several starting configurations with different volume fractions from 0.8 to 7%, so supersaturation conditions, to be able to observe quite a number of clusters in a 100 nanometers cubic box. So there are some specific parameters for Brownian dynamics. The Brownian dynamics friction coefficient, the mu, which is the dynamic viscosity of water at the simulation temperature, and tau t, which is the time constant for coupling. So the molecular dynamics simulations run in an HPC facility for several months before obtaining few large clusters. The computational time depends mainly on the number of particles. Titanium dioxide nanoparticles start to aggregate within the first trajectory frame, so in few nanoseconds, in agreement with experimental observations. In fact, titanium dioxide nanoparticles in suspension are commonly coated by polymers or silica or aluminum to prevent flocculation. But how do we get descriptors for the aggregation process? So it is interesting to see the speed of the clustering process. So we can plot the cluster number in time for the different volume fraction and particle sizes. We can see the cluster size distribution, which is the cluster number versus cluster size for each volume fraction. And we can also see the effect of the initial nanoparticle size, plotting the cluster number versus the nanoparticle size for each volume fraction. The very first thing to do is to find a way to identify clusters during the trajectory. We employed the unsupervised machine learning algorithm called dbScan to divide the nanoparticles into clusters based on a distance criterion. Over other unsupervised algorithms, dbScan has the advantage of not requiring the knowledge of cluster number a priori. Instead, it is able to divide the particles into clusters based on a pure distance criterion. Of course, the hyperparameters must be tuned to get the desired information on clusters. This is done using a try and error method, basically running the algorithm with different hyperparameters and visualizing the output like I show here in this slide, where I used the matplotlib Python library to plot the resulting division in clusters obtained with the tuned hyperparameters. So basically there are two hyperparameters, epsilon and also mean samples. So here we see the speed of the clustering process. So I plotted the cluster number in time for the different volume fractions and the particle sizes. You can see that the aggregation takes place very quickly for pure titanium dioxide anatase in water. Clusters form already within 2.5 microseconds with the most concentrated solution, so the highest volume fraction, showing also the highest clustering rate. On the left, the 3 nanometer nanoparticle, and on the right, the 4 nanometer nanoparticle. Despite the volume fractions are the same, so in the case of the 4 nanometer nanoparticle, there are much less nanoparticles in the box. But the aggregation is slightly faster in the case of larger nanoparticles. The cluster distribution, which is the cluster number versus cluster size for each volume fraction, and I choose to take like a snapshot after 1 microsecond of simulation. Here the data for the 4 nanometer nanoparticle is plotted after one microsecond of simulation, which is a time where there are already many clusters in solution, but the clustering process is not yet convergent. I mean, there is not yet a single final large cluster. Quite obviously, in the most concentrated solutions, nanoparticles form larger aggregates of up to 700 nanoparticles in the 7% volume fraction, whereas in the lowest concentration we get clusters of 2 to 10 nanoparticles. The effect of the initial nanoparticle size is shown here. So basically I plot the cluster number versus the nanoparticle size for each volume fraction. You can see that the final stage of the clustering process is reached more slowly in the case of small nanoparticles, but in any of the considered scenarios, the clustering is taking place within the first 2.5 microseconds, and few modifications are observed thereafter. The larger clusters merge and change slightly their shape, basically. The final large aggregates are mostly spherical or elliptical, and some of them have branches. So, to summarize, we employed classical molecular dynamics simulations with the umbrella sampling technique to get the free energy profile for the aggregation of 2 identical nanoparticles in explicit water. We fitted the repulsive short range and the attractive long range parts of the curve, and we built a table potential we then used for running Brownian Dynamics for at least 30 microseconds of trajectory. We analyzed the Brownian Dynamics trajectory using an in-house Python script which implements the dbScan clustering algorithm to find clusters in each trajectory frame. So this represents a multi-scale modeling scheme that provides a number of descriptors which can be used in QSAR models to help predicting nanoparticle toxicity. So thank you very much for your attention, and you can ask questions during the Q&A session, and also feel free to drop me an email if you have any doubt on this topic. And thank you.
Hi, my name is Buchete. I'm an associate professor in University College Dublin, the School of Physics, and we are part of Word Package 3 in our nanoinformatics project. And I will tell you about our attempts to model protein-protein and protein ENM, engineered nanomaterial interactions in general, but nanoparticles in particular. And we want to calculate descriptors that tell us the biophysical properties, quantify the biophysical properties of proteins interacting with nanoparticles. So as we know, for more than a decade now, once nanomaterials and nanoparticles in particular enter in contact with biological material, they have particularly favorable interactions with a broad range of biomolecules and proteins in particular. So they get covered with with proteins, and have been proposed several approaches to quantify this process have been proposed, and it is understood that there are molecules that bind proteins that would bind tightly to the nanoparticle surface, for example, and we call them, according to the literature, you have a review here at the bottom, suggested hard corona, and the softer, less well bounded molecules on the outer layers, they form what's known as the soft corona. So in our attempts, we are trying to model not so much the kinetic process of corona formation, but the equilibrium properties of the hard and soft coronas of protein covered nanoparticles. And it's important to realize that for some of the proteins of the nanoparticles that we can study in detail in our work package with the methods available, starting from quantum DFT calculations all the way to atomistic molecular dynamics, molecular docking, and coarse grain simulations, as you will hear in the next talk as well. We are dealing with nanoparticles that are relatively small, right, compared to the broad range of nanoparticles that are used in experiments and available. So nanoparticles that are between 2 and 4, 5 nanometers in diameters will have about the similar, right, similar sizes, similar diameters with some of the proteins that we are interested. We know that they participate in corona formation in different biological environments. Mucin is an important protein, the human surfactant protein HSP. D, for example, human serum albumin is a protein that we have been using in our simulation. Antitrypsin, apolipoprotein is a smaller protein that is also important from a biological point of view. So these proteins are illustrated here in this figure, and you can see that they are all about the same size with some of the nanoparticles that we are using, and I need to acknowledge here our collaboration with Professor Monica Catayud from Sorbonne University, and the Polito group that you've just heard the previous talk about. That um, so we are using some of the nanoparticles that they are using and developing as well. So our idea was to, on one hand, put together experimental information that we may have about protein structures of proteins of interest, and um, calculated experimentally relevant, let's say, information of nanoparticle structures of engineered nanomaterials. And some of them, this is a complicated issue, some of them may have different types of coatings. In the studies that I'm going to illustrate here, we are considering just pristine, not covered nanoparticles, bare nanoparticles. And we want to put it together, the information from these two types of sources, and predict something, be able to calculate the biophysical properties of covered uh, nanomaterials and nanoparticles in particular. And one way to achieve that, so there are several ways where one can go about it, is to use molecular docking and generate models, a broad range of hopefully representative models of proteins covering nanomaterials, right? So after the docking step, we want to analyze and use right this broad range of models of coronas that we are generating to calculate biomolecular descriptors such as the hydrophobic fraction of the solvent accessible surface area. So I'll use in this talk my abbreviation for this descriptor SASA, right? So SASA is the solvent accessible surface area that is a important biophysical descriptor that tells us about how hydrophobic in this case, or hydrophilic, or charged, positive, negative, the protein covered nanoparticles are. So this is useful information that right, similarly with the other descriptors that we are calculating, can be used in QSAR approaches to make connection with nanotoxicology. Is one of the goals of our the main goal, right, of our project. Right, so this is the brief description of the pipeline of our approach, and this is very simplified, right? As we know, both the nanomaterials, but particularly the protein structures, they can be very diverse, and they need to be prepared, right? So in order to get more realistic description of our proteins, which are dynamic quantities, I want to emphasize that, so people are very familiar with frozen X-ray derived structures, X-ray crystallography derived structures of proteins that are available in protein data bank databases, for example, right? But in physiological conditions, proteins at room temperature, at physiological temperature, they are dynamic objects that change their structures and fluctuate. And while we have access to detailed information about their atomic level composition, right? We also want to find information and to include information about the structures that proteins can adopt. And these are some movies here that I'm just using to illustrate very briefly the molecular dynamics approach that we are using first as a first step to study the dynamics of these proteins and find a broad range of conformation that they may have. Without getting into much detail, just because this is a school, I'm mentioning here that we generate these movies by using what's called the molecular dynamics program, and in particular we are using the NAMD program, which is very very popular and easy to use with lots of tutorials with the latest Charm 36 force field. And here is just a brief representation of the interaction, it's a truncated version of the main interaction forces of the spring type for the bonds, quadratic potentials for certain angles. We can also have dihedrals, which are representing as illustrated here, the relative conformation, relative positions of atoms, of each and every atom in our protein. And we have non-bonded terms of the Lennard-Jones and the electrostatic type as well. Right? There are other additional terms, but it's not the scope of my talk to get into much detail. But with molecular dynamics, with atomistic molecular dynamics, we can take from the protein data bank some of the structures of the proteins that we are interested in and investigate using relatively long molecular dynamic simulations what are possible conformations that are adopted, and use them in a statistical way to sample the possible bindings, right? Because different binding, want to look at protein-protein interactions, and one may expect that different conformation may have different binding propensities. So since right, it's very expensive to take into account when calculating this description the full conformational dynamics flexibility of proteins, which we can do. So part of our research is to use Markov state models or milestoning, which are enhanced sampling methods. So methods that allow us with relatively with reasonable resources, computational resources, to calculate the conformations of proteins. We can also, I'm illustrating here, this is the root mean squared deviation versus the simulation time for 3 of these proteins that are shown here. And we can extract outliers, for example, so we can have a range of conformations that we can use later on to probe how diverse the binding properties, right, without doing full flexible docking, how diverse these binding properties in docking can be. So the next ingredient, as I was mentioning earlier, is to use a docking program, and we tried several of them. We are using several of them. PatchDock is a very good one that I'm recommending. You have the references at the bottom. ZDock is also another good program. So what does it mean, a good program? Well, it has to be we have to be able to run it quickly and efficiently on relatively large systems. So typical molecular docking programs have been developed to find best docking poses of relative positions of relatively small drug molecules with respect to a big protein. Right now we are using basically docking, as in the second illustration here, to um, to dock big proteins with each other and with respect to nanomaterials. And these are examples of what we are getting when using this approach. We can build models of protein covered nanoparticles. These are 4 nanometer titanium dioxide. I will tell you in more detail on this slide that where we can attempt to dock in various ways in order to get average values, the different conformations of our proteins on these nanoparticles that can be of different diameters and different shapes, and right from different sources, and calculate the solvent accessible area, and in particular the hydrophobic fraction, for example. And this is what we obtain. I will use this graph. So as a function of the number of proteins that we are docking subsequently, right, on different nanoparticles. And this slide summarizes results for both titanium dioxide and silica nanoparticles, right? So on this axis, right, so our results for adding more and more proteins to the corona, and here is the value of the accessible of our descriptor that we want to calculate. We can just do it without any nanoparticle, right? We can just look at homo or hetero oligomers of these proteins that are docked together and see what trends do we get. And we can estimate and calculate, describe quantitatively what results are we going to get if we dock different number of proteins in different combinations on titanium dioxide, for example, or on silica nanoparticle, and we obtain here different trends. So for silica, we have these dashed lines here that show the trends obtained in this case, and for titanium, right, I highlighted with ellipsoids here the data points. Right, the diamonds here correspond to the diamond-shaped nanoparticles that we've seen in the previous slide. So we can go even farther and calculate more detailed molecular descriptions of our corona protein-covered nanoparticles and focus, for example, on charged patches, either from an average point of view, what is the average SASA value, right? Average solvent accessible surface area that is positively charged or negatively charged for titanium dioxide. And similarly, you see that we have, instead of 40-50% as we had for the hydrophobic fraction, we have much smaller values of 12% and 14% maximum, and for silica as well. And here I'm just showing a lot of data that my students are putting together in using different types, considering different types of nanoparticles with different diameters. And I will need again thank our collaborators for providing those for us. Right, so to sum up, right, I showed you how using this approach, using molecular dynamics, refinement of protein structures, and generation of docking candidates, and in conjunction, right, with docking programs that allow us to look for realistic poses of proteins on nanoparticle surfaces and score them and rank them according to the scores that come out of the docking function. We can describe these interactions, and we can zoom in and describe particular interaction between, let's say, particular proteins and particular nanoparticle surfaces as illustrated here. Right, we can then compare our results with similar results from coarse grain modeling, which is more expensive. So docking itself is a coarse grain method, but right, there are other more expensive coarse grain modeling approaches that one could use for, and we are collaborating on this with the next speaker, and also with Vladimir Lobashkin, from my colleague from UCD. We are using machine learning to upscale our results, and we want to predict, for example, to use this to predict the effects of perturbations such as mutation and changes in molecular composition. And I will finish by acknowledging Vinu Anapurani, who is the main student, my PhD student that has been driving this work. Agia was a master student that contributed to our first efforts, and again my collaborators Vladimir Lobashkin from UCD and Monica Katayud, and of course the speakers in this session. Thank you very much. We are looking forward to the discussion, the questions and answer session. Thank you.
In the next 15 minutes, I'm going to talk to you about coarse grain physical modeling and how that can be used in nanotoxicity, and about some of the results that we obtained in the context of nanoinformatics in our project in Work Package 3. My name is Agur Sevink, and I'm working with Ankush Singhal, my postdoc in Leiden University, in this project. Okay, before we go to the modeling part, or the coarse grain modeling part, I first want to talk about what is really modeling, because in different fields, the concept of modeling is different. So in chemistry and biology, a model is usually a visual conceptual representation of measured facts. So you do a measurement, based on the measurement, you generate a model. And this is usually very dynamic. So it changes if you have more or better measurements, which basically gives you more insight in the phenomena that you're looking at. In information theory, like machine learning and QSER, modeling is using descriptors or correlations for interpolation and optimization. And it's also usually a dynamic process. So the more and the better the descriptors for more materials you get, the better your prediction by means of interpolation or optimization can be. In biophysics, the situation is slightly different because you base your model on physical laws. So models are usually formulas which describes you something in terms of something else. It's more or less static. So once you have a model, the model will always give you the same result, unless you change the model by including more phenomena or by considering higher precision. But as it stands, the model will always give you the same. So it's good to keep this in mind if we're going to discuss the models. So starting with the most basic level, ab initio quantum mechanics, you can describe molecules in terms of their wave functions and their nuclear coordinates. And this is just an example of the molecular orbitals of a single water molecule. And the first four to the left are the occupied orbitals. The fifth is not really an occupied orbitals. But this will, on a general computer, even this calculation of these molecular orbitals for a single water molecule will already take you quite a lot of time. So what if you want to look at collection of molecules? And then usually for a very big simulation, for a big calculation, you go to continuum mechanics. So this is a calculation or a simulation for the flow field of very many water molecules through a certain sea arm in Holland. And you can even look in time, so it will give you the current of water molecules. But compared to the quantum mechanical description, you lose all the molecular information. You lose also the electronic information, so the electrons, but you also even lose the molecular information. So you just see the result of very many water molecules flow. Okay, so if we are interested in physical modeling of a certain phenomena, we first have to consider the relevant scale. And the relevant scale in nanotoxicity, or in general in nano devices, is, let's say, 10 to 100 nanometers. So it's between an antibody and a virus. So we'll immediately realize that if we will want to consider quantum mechanics, where there are basically no parameters that we have to come up with, we are not going to make it, because quantum mechanics is very compute intensive, so we can just do a few molecules. But if we go to the continuum mechanics, where you can describe the behavior of a tennis ball, for instance, or even larger systems, then you lose the molecular resolution. So that is also not what we want. So we have to come up with something new. And how do we do that? Generally, we do that by coarsening. So you start with ab initio, you have a quantum mechanical representation, and you can cast that into a classical atomistic representation. So you cast all the effects of the electron cloud on the nuclei and the bonds and so on into effective potentials, so torsion potentials, Leonard-Jones potentials for the unbounded interactions, and also the angle potential, the bond potentials. And the idea is that you retain the shape of the underlying free energy landscape. So the quantum system has a certain energy landscape over which the system can develop. And the idea is that the full atomistic representation will kind of retain the shape of this free energy. And you evolve in quantum calculation, you evolve by the Schrödinger equation from continuum wave equations. And then if you have a classical representation in terms of an atomistic force field, you just develop the system in terms of the Newton's equation of motion, which gives you the force is m, the mass times acceleration for the discrete atom positions. Okay, so now we have a representative atomistic representation of our system. Is that good enough? So if you consider the systems that we are interested in, which is in the order of 10 to 100 nanometers, and often also the time scales of nanoseconds to seconds, then you will immediately realize that also the atomistic description is way too compute intensive. So on the left here, I have the atomistic representation of a lipid in this case. So I use lipids as an example. So what you can do is actually take that atomistic representation where you describe all the atoms by a particle and condense a number of heavy atoms into a blob, basically. So the second one, you have condensed the full atomistic representation into 15 sites, which are just blobs. And you can even go beyond that. You can represent it just by a head group or a tail group. You can represent it by two blobs, so just a head and a tail bead, or you can just represent it by an oval shape, which has a director. And what you see is that by going from left to right, you increase the system size and the trajectory length by reducing the number of coordinates that you have to trace. But going from right to left, you decrease the chemical or structural detail. So it's just a balance of those two. Okay, so in CG, as I mentioned, you have a reduction of degrees of freedom. So the degrees of freedom are the positions and the velocities. So if you have less coordinates that you trace, you are basically more efficient, but you lose the chemical resolution. And another effect is that if you coarse grain, so if you represent your system by less and less degrees of freedom, you have a softening of the interactions, which allows you to take a larger time step and to simulate longer trajectories. So these two basically make CG descriptions very effective. Then about the descriptors. So you can also calculate different descriptors. So in the quantum level, you can calculate the electronic descriptors like the homonumal gap, surface defects, dissolution, charge distribution, and so on. At the atomistic level, you have the atomistic descriptors. So they're generally geometric, energetic, effective charges, coatings, and so on. And on the coarse grained level, which contains actually many different levels depending on the coarse graining level that you choose, you can have the system descriptors. So you can have binding energies, corona effects by solution or the binding of proteins, coagulation, and nanoparticle-induced remodeling of membranes, for instance. So what you see is actually it gives you a range of new descriptors which may be interesting for machine learning or QSAR type of developments. And what you can also see is that the lower level, so the QMD, AA, the all atom MD, give you information about the nanoparticle, and the higher level, so the AMD and the CGMD descriptors give you an idea of the nanoparticle environment interactions and also some of the mechanisms. Okay, so now I turn to the system that we have been studying. So we are basically interested in nanoparticle inhalation. So what happens if you inhale a nanoparticle? And on the left, there is a picture of the alveoli, so the lung kind of zoomed in. And then if you zoom in even more and you look at the interface of the alveoli with the air and the alveolar fluid interface, you see that there are lipid membranes like everywhere in our body. But these lipid membranes are quite particular. So what we want to do is to represent them in a physical model. And that is actually what we did. So we selected a representation first on an all atom molecular dynamics level of a model lung membrane, which consists of a number of different lipids, the PPC, the OPC, POPC, and cholesterol in these fractions. And we used steered all atom molecular dynamics to calculate actually the binding energy between a nanoparticle and a membrane. And that is depicted on the right. So you set up a system and you slowly pull the particle towards the membrane, and you basically measure the thermodynamic force exerted on that nanoparticle. And if you do that right and use a Brothers sampling, you can calculate the binding energy. So the binding energy is actually the energy that the system will gain by going from the solvent, which is in the plot on the right side, so far away from the nanoparticle. On the horizontal axis is the distance from the midplane of the membrane. And if you get closer to the membrane, it's actually attracted to the membrane. So you see a potential well, and by measuring the depth of this well, you can actually determine the binding energy. And we did that for 3 nanometer silver nanoparticles, for 5 meter silver nanoparticles, which is actually quite close to the limit of all atom molecular dynamics. And we did that for silicon oxide, for titanium oxide, and we also looking into C60 and carbon nanotubes. So if we want to get to the experimental sizes, where the particles usually have a diameter of about 20 to 50 nanometer, we have to use coarse graining. So what we did is we used an existing coarse grain molecular dynamics representation, where you map 4 heavy atoms to 1 CG bead. And we did a trick actually to separate the particle in a core and a shell part, where the core and shell is actually not different materials, but they have slightly different properties. And there is some work of Tam et al. Who described the descriptors for these in terms of these core shell representations. And we use that to basically fit our parameters in the coarse grained representation to the fine grained representation. And on the right, you see how the coarse grained potential mean force that we calculated using kind of different parameters for the core and shell region matches to the atomistic potential mean force calculation. And in fact, only the depth of the well matters, and not really the precise precision. So what you see is that we are able to reproduce the full atomistic potential mean force calculated with the coarse grained calculation. And then that is what we did. So then we started from full atomistic for 1, 3, and 5 nanometer particles. And then we continued to the 10, 15, and 20, and even larger nanoparticles, which we would not be able to do with the atomistic simulations. And then what we see is that depending on the size, we get different binding kinetics. So for 1 nanometer particles, you basically get permeation. So the nanoparticle is much smaller than the membrane thickness. So it's behaving like a molecule which just permeates through the lipid membrane. 3 and 5, where the size is comparable to the membrane thickness, which is about 4 nanometer, you see wrapping. But if you go to larger nanoparticles, you see a wrapping, but also a budding. And the question is, okay, if we go to even much higher or much larger nanoparticles, what will happen? But we have an idea that the kind of 15 is a size which is representative for the larger nanoparticles. So what we did is we used constraint MD to calculate the binding energies, as I showed you before, which relates to the hydrophobicity. And we used unconstrained molecular dynamics to determine nanoparticle attributes, such as the solved accessible surface area of the nanoparticle, but also the nanoparticle-induced membrane remodeling effects, like the formation of membrane defects, the lipid diffusion and partitioning, and the raft formation, and so on, which are basically particulars of the environment and not necessarily of the nanoparticle, which is an added bonus of these coarse-grained simulations. So is the membrane important? So can you attribute everything to the nanoparticle? And this shows you that actually that is not the case. So if you look at a lung membrane and you take the same particle and you add it to an asymmetric model plasma membrane. So in the lung membrane, the lipid composition is symmetric over both leaflets. In the plasma membrane is asymmetric, and what you see is the effect on the membrane of the nanoparticle is quite different. So this is also an attribute of the nanoparticle in terms of the effect on the environment. Okay, the next things that we are doing and planning to do is to also look at small molecule absorption, and we compare the binding energies from the all atom molecular dynamics PMFs, the potential mean force that we calculated, quite similar to the nanoparticle membrane interactions. And we use those predictions or these calculations to compare to QSCR predictions. And we do that together with work package 4. And we also are intending to add protein descriptions. So to have coarse-grained proteins and to add them to the nanoparticles and see how that affects the binding kinetics of the coated nanoparticle with the membrane. And something that we're also looking at is going to even larger and longer. So there is a kind of hybrid discrete continuum method, which even kind of coarsens beyond the Martini model that we considered. And this is an example of which looked at carbon nanotubes. In solution, they bundle in these kind of bundles. And the question is, okay, if these bundles meet a membrane, how do they interact with the membrane? And actually, this is a simulation of a quite long bundle of 20 nanometer long, interacting with the lipids and actually extracting lipids from the membrane and thereby destabilizing the membrane. Okay, so I hope I've given you a kind of overview of what we are capable of, and I thank you for your attention.